thanks so much for joining. It's very nice. We will try to have this meeting more often, like every month or every 2 months. We'll try to accommodate also the Australian people because that's not convenient for them, obviously. So that's something we've been preparing with Léa, Ilia, Frank, and also Pierre-Lune Bellec, who joined the team. So he is a researcher in Montreal. And I don't know, Lune, if you want to just introduce yourself in 2 minutes.
So yeah, I'm based in Montreal. I've been involved in fMRI method development for about 20 years, and in denoising methods in particular. So I used to work with Clara during her PhD, and I'm really delighted to join this very active group. So hi everyone.
Thanks a lot. So yeah, so the agenda, we send it to you by email, but basically we want to give you an update on new things that have been implemented in HALFpipe. We have some update on the guidelines that we would like to discuss with you. We also submitted a R1 in February last month, so we will give you an update about that. I mean, we don't know anything, but just what we submitted, and we will have some time for questions. Okay, so for the update about HALFpipe, so one thing that was missing, like last meeting in Amsterdam, was scrubbing. And in the survey we did with Léa, people like you all say that it would provide benefit, or even it's required to have scrubbing in our analysis. So that's why originally, when we designed the guidelines with all of you, we said that we will have 2 pipelines with scrubbing. So until now, it was not available. And so Ilia's team has been implementing with Frank's team scrubbing in HALFpipe. So now you can use it. So meaning that basically everything is ready. So you can start your analysis with scrubbing. So that was the main issue that we had last time. We were waiting for that. The second issue was the fMRI prep version. So we asked you that also in the survey last year with Léa. Until now, HALFpipe was running the LTS. So the version like from 2020 that was maintained by Pierre-Lune's group, actually. So we ask you if we should like update regularly the version of fMRI prep. So a third of you said we should implement new version that would be better. So that's something we have been working on. So we are now moving from the LTS, so 20.2.7 to the 25 version that has been released on Wednesday by the fMRI prep team. We met with them last week, and we are presenting and discussing with them about like HALFpipe issues and the fMRI prep implementation. So they are very helpful, and Léa and I and Lune, we are going to join regularly the meetings with the fMRI prep people. So we will be up to date. So it will help to have something up to date for HALFpipe. We also have Bertrand Thirion with like Nylearn and scikit-learn lead developers that agree to help us advising methodological and implementation decision. So thanks to Pierre-Lune. So it's very nice to have these people on board with us. So this new version of fMRI prep has been implemented in HALFpipe. So if you download today the dev version, it's here. My students and Frank's students have been extensively testing it in the last 2-3 weeks. We are correcting bugs every day, kind of. So Léa corrected 1 bug today, actually, with a core implementation. So now, for example, we compare the result. So that's the 3 pipelines we've been asking you to use. So basically, pipeline 1 was core, pipeline 2 was motion parameters and scrubbing, and pipeline 3 is motion parameters, scrubbing, and GSR. So we compared, here's the correlation between the LTS version of fMRI prep into HALFpipe and the new release of fMRI prep, so the 25. So that's for row connectomes, and that's for like case control analysis, beta matrix. As you can see, it's pretty good for pipeline 2 and 3. It was not great for pipeline 1, but we figured out why. It's about the implementation of ACOMP that differ between both versions, but Leah fixed it. So now we're good. We have to rerun the analysis. So as you can see, we are still like finding some bugs, but we are almost, I mean, it looks like we are almost here. So we are still going in the coming week to test that the implementation is perfect. You can use it if you want. It's on the GitHub. But what we want is like in maybe 2 or 3 weeks, we will send an email to say like, I mean, and Leah will probably send an email to say like it's not a dev version now. It's it's a new image of HALFpipe you can use with the new fMRI prep LTS kind of. So that was for fMRI prep update. Leah, do you want to add anything about that?
Not much. I mean, I think yeah, one of the things that Violetta from Ilya's team and I and Ilya are looking into is also like, because the 0.9 is not a perfect correlation, right? So we don't really know what this means in terms of like differences to the outputs in terms of scientific differences, right? Will we find something here that we wouldn't find in something else? And so on, like, would this actually impact scientific results? So one thing we're also looking into is this kind of stochastic variability. If we rerun just randomly, because there's some variation to results from just randomness when you run a pipeline multiple times. So we believe that the 0.9 could potentially just be because of this kind of stochastic variation when you rerun stuff, but we don't know it yet. So that's also something that's going to be a bit of an investigation, and we're honestly surprised by the amount of variability that is in the processing. So that's going to be something that's going to be super interesting that I hope to update you guys more on in the future. Yeah.
Yeah, so we have been working on that. So for example, if you like, my students have been rerunning a data set several times, and they compare the result like for the same data set. So the correlation is high, but it's not 1. So we still want to figure out like this. I don't think it's going to impact your analysis anyway, but we are doing a lot of sanity check like that to be sure that the version you will use is stable, and we want to understand these differences, obviously. So that's why we are talking with the fMRI prep team, if it's something about HALFpipe or if it's something about like the fMRI prep new release, because obviously between the 20 and the 25 version, they made a lot of changes. So we want to quantify that, but we are close to be done.
So Clara, just real quick, Dr. Van Orp has a question. Will IC-Aroma still be available on HALFpipe? And it's not going to be removed from HALFpipe. IC-Aroma would still be available. Sorry, I was in the chat. Just want to make sure we.
Oh yeah, sure. Everything is going to be still available. It's more like we are giving you recommendation, and also if you want to use the LTS from like of fMRI prep, that's the 20.2.7 or whatever, you can still use it. We are just giving you the new version, but you do what you want. We give you recommendations. Then we have so Ilya in Amsterdam presented the new UI. So I don't know, Tomasz, if you want to say a few words about it. You have been developing it.
Yeah, basically, Leah asked me if I can like show short demo. So yeah, if you are then done, just say and I can show like and say a little bit more about it. Yeah, I can do it also right now. But you have to stop sharing, then I can. Okay. Okay, so yeah, hi everyone. For those that don't know me, I'm Tomasz from Ilya's group in Amsterdam, and I was working mainly on the new UI for HALFpipe. So basically, this is how it looks like, and I will walk you through it now, just to show you like how it's structured and how it works now. So it basically copies the old functionality of the old UI, but in a very different graphic layout. So you start by selecting the working directory. So yeah, I will just paste here so that it's faster, but otherwise you can use this browse, or you can just type in or what you like. Then the input data tab is here. Here you select the data, whether it's like bits format or not bits format. I will use now the classic bits format, and later I can just show you also how this works with non-bits file format. Okay, so you select where is your where are your data, and yeah, then suddenly you see that you get like more tabs here, and this is basically saying that you can start to make your configuration for the pipeline. Here are like general preprocessing settings. Okay, whether you want to turn on slice timing, then you are prompt if you want to use these values. So for those that use the old UI, they will instantly see the similarities from these modal windows. And also, like, these modal windows are movable, so if something is obscuring information under it, you can also like move it. And yep, and you make some weird selection. And yeah, there are also some further configurations that you can do, and also advanced settings that I will not show now. But here are the features. So basically, you go to do a new feature, and here you have the selection menu of the type of your feature. So let's say you want to do task-based. You select name, and then you will get this whole form of your selections. So first you select some of the tasks, and you see that you will also get now that you can select contrasts for the selected tasks. And also here you see now, based on the selected task, how many both images, image files were found. And you can also like open and check whether this makes sense. And okay, if you want to add contrast, you just click add contrast, just select some name and place some values and confirm, and it will basically add here columns in the table. If you just add another one, it will just add here another column. Then you have here individual preprocessing settings, like if you want to use smoothing, value of it, grounding, scaling, etc. So basically, you just, if you want to change this, you will just click in the window and change this. And yeah, you can also change the type of the filter, you can also change these values and so on. Here you have this panel with remove confound. So if you want to select some of these functionalities, you just click the checkbox and you are done. Yeah, and similarly, you just add another feature if you want to continue. So, for example, this one, and then you will get a different form based on the type of the feature. And all of the features are grouping in this panel here. So if you have everything that is Rio type, for example, will be here. All the task base are here. So these are like collapsible menus here. And also you can copy the form. Here you have the button for duplicate, and it will now create a duplicate from the first one. So yep, and okay, going further. Here you have tab with the group level models. So the layout is the same, but now we have two options: intercept only or linear model. So let's type in name. So we will get some choices. For this, it's very brief because for this type you don't have that many options, but for linear you have much more. Especially you have here, you have to add first the data spreadsheet. So you add the covariate spreadsheet. I can just paste path to it, and then you will get some settings of the file. So you have to set based on the data inside the spreadsheet. You have to just assign which column is the ID column and which column contains the continuous or categorical data. So let's say these two, you confirm, and then you will get like further selections here, and you can make again some choices, selections. Also here is again if you want to add additional contrast for this, and this open similar tables like it was shown before. Okay, set some values. Okay, so basically more or less if you are done, then you go to check and run. You can press here refresh, and if you forget, for example, to select the task, you are warned about this. So for example, we didn't select any task here, so we select it, go back, refresh, and you can basically this what you see is the spec.json file. This is all the configuration that will HalfPipe use, and you see that all of the choices that we made in the forms are basically captured here in this spec file, and then okay, you can run the HalfPipe. So this is basically how it looks like, and yes, you then can quit if you want to quit, and I will show you just two more functionalities. Will be this will be short. If you, for example, have an existing spec file, you can just again find the path, and if this UI founds an existing spec file in the working directory, you are warned if you want to overwrite or load. And if you basically click and load, it reloads everything from the file, all the choices that are. So basically, you see that this regenerates all of the widgets with all of the choices that are inside. So yep, then you see again it's still loading because yep, always when I share this via Zoom it's a little bit slower, but otherwise it's quite responding fast. So yeah, it basically you see it loads all of the things that are in the spec.json file. So this is second thing, and the last thing that I wanted to show you is the case of the non-bits file. Just I will show only the first tab with just to give you an idea how this works. Okay, so if you have non-bits data, then you turn this switch off because you don't have bits data, and then you have to add the T1 weighted images. So you can also copy paste here, but now I will not do it because I want to show you how it looks like normally. So you can navigate to your files, and you select some of your T1 files. Okay. This isn't correct, but yep. otherwise I will just copy it here. Okay, so basically you just select the text of the, this is like wildcards of the file names. So it just basically says where is subject in this file name, and then you can okay, submit. You see like how many files were found, and then you just click OK, and then you do the same for the old files. So you just put it here, you see how many were found, you just click OK, and then you confirm, and your data were loaded, but you get a warning because you didn't set the working directory. So you would just go back and set the working directory, and then you see, just like before, all the remaining tabs here. And yeah, since this point the work is similar. No, it's the same like before. Okay, so yep, this is in short how this works. We are still like testing this within the group, and yeah, once we are done testing within the group, we will just let also the wider group of users test this and get some feedback to tweak this more, so that at least majority of people are happy with how this looks like and works.
Thanks so much. Yeah, something that I've been asked in the chat, like you can still use the old user interface, and it's still going to be the default, but you will be able to use the new one just by specifying it in your code in the terminal. It's still the terminal running. So yeah, it's just like a general question.
So basically for now it's default is the old UI, but you can use the flag TUI to run the new UI. But later, once this is like fully tested and we are like sure that it really works, we will just make this a default, and with time we will abandon the old UI and just really move on to the new one.
Yeah, maybe as a quick aside, I really want to highlight how amazing it is that we have these windows you can move around in the terminal. So you SSH to your cluster and you have these windows, you get immediate feedback, you see the files that are being selected. So it's kind of, we hope it will solve all of those problems with like many errors that you get later on by getting all of this immediate feedback, that it will make using HalfPipe much easier and much more approachable. So I'm really happy with how it's turning out, and I hope that we can release it to everyone very soon. Thank you, Tomasz.
You're welcome.
Raj, you wanted to ask something?
Quick question. Okay, dumb question. The interface you showed just a minute ago, that's the new one or the old one?
Yes, that's the new one.
Old one was just aligned in terminal with questions.
Mm-hmm. Oh yeah, now I remember.
Any other questions? No? Okay. Okay, so
I was going to put this in the chat, but we're putting together this guideline sheet now for how to deal with common issues like skull stripping or cross-registration not working, and how to correct that in HalfPipe. And right now, that's a lot of manual editing of spec.jsons and a bunch of manual coding. Is there any thought to wrapping some of that into the UI?
So HalfPipe is an open source project, and I think I'd be super happy to accept any of those special scripts into HalfPipe, and we can definitely collaborate on making that happen. Maybe we should circle back to that in a separate meeting. Greg, should we schedule something via email maybe?
Yeah, let me send you what we're coming up with, because right now it's an algorithm. I was just wondering if you're incorporating any of that sort of debugging world into the UI as you're writing it.
Not yet, but it sounds like a fascinating idea. So let's keep in touch about that.
Okay.
Thanks.
So then we have like some general update about the guidelines. So yeah, that was the previous version that we sent to you all. There was some discussion about motion criteria. So until now, we recommended to do a visual QC and a mean frame-wise displacement under 0.5 millimeter. That's something that is vastly done in the literature. We were quite comfortable with this threshold. We still recommend to add a max FD under 3 millimeter. I know there was like some emails about that in the last month. So we can rediscuss that with you. But yeah, we were thinking, I mean, we had specific meetings about that with the different chairs, but we're thinking it's a good compromise for now. Something that we have to keep in mind is like, it's done after running everything. So it's just like removing more subject. So something that is always obviously better is like if the different sites are able to share like individual level derivatives, we can easily remove more subject, like using a 0.25 threshold mean FD. So that's obviously always better. But if sites are sharing like group level analysis, then we will not be able to redo it. Basically, you do not have to rerun HALFpipe. It's just like when you do your case control analysis that it matters. So it's going to be easy a posteriori to rerun your analysis with another threshold if needed. But we still think for our guidelines, we wanted to have like some like this threshold, which is quite accepted by like the general community, but happy to discuss with you if you have questions about this.
Yeah, we have been finding the frame-wise displacement of less than 0.5 millimeters rarely is exceeded in functional data. It's fine to use that. You're just not going to reject very much. The frame-wise displacement, the max being less than 3, basically says that if anybody moves their head on even one frame a bit and moves it right back into position, you're rejecting that whole run. And that is far more stringent than most of the groups that we've seen. So I'm recommending, like some number of frames, like we're doing 25% frames outside X displacement or something like that. But I wouldn't use the max frame-wise displacement criteria at all, because we were rejecting far too many scans based on that.
So I don't know if anyone else has feedbacks on that. Lynn, do you have any idea about those thresholds?
I would tend to agree with the feedback that it is more of an extent of motion. I think there's quite a bit of data to show that motion is a fairly consistent characteristic, more of a trait than a state. And I would expect somebody who exceeds 3 millimeter to exceed 3 millimeter repeatedly. But in principle, I'd agree that if there's a single motion of high amplitude in their series, it would be a shame to throw the data out.
Hi, this is Guido from Amsterdam. Just my thoughts is that typically if we use clinical scans, especially from kids, that you get quite some movement. Consider like ADHD or so. So if that's also your aim, which probably is, at least for some consortia, then I wouldn't explicitly put it in anywhere, that it's but just leave it out and you can decide afterwards. So you should not be necessarily rerunning things to set a different threshold for QC. So just log it, and dependent on your research question, you can be more stringent or not, because if you have to toss away like 50% of your participants, you get nice scans, but it will not be representative. And if you have a different type of sample where people like regular adults without any of those symptoms can just lie still, you can be more restrictive. So I would not set a threshold across the board for everything.
Okay, thanks so much. Yeah, any other point of discussion for that?
I would also agree that the exclusion step is only done at the second level. So specifying a file with the filter makes sense to me. So there is then still a flexibility to control that.
Yeah.
Absolutely clear. The other question is if those two criteria here would be the main guideline points. Is that correct, or do you have other say revised recommendations for the more visual based QC steps that I found more tricky to decide in some cases? Not necessarily the ICA aroma thing, that's a topic of its own, but the carpet plots. Do you still keep up the recommendation to decide about inclusion or exclusion based on the carpet plots beyond these two numbers?
I think we do recommend to use the carpet plots for visual quality control, and the new FMIPIP version has also updated them to try to make them a bit more clear. So they now have more clearly ordered sections, and they also order the voxels in a way to make kind of patterns more clearly visible. So in theory, it should be easier to decide for the carpet plots with the new release. So with respect to the motion cutoffs, I think, yeah, this is very much a gray zone. And I think this was also where one of Ilya's PhD students in Nadja. So one of her jobs was also to kind of rework the QC manual. So that's been kind of on our list for quite a while to kind of incorporate all the information we've been getting from sites and those kind of problems into some more updated criteria. But unfortunately, there hasn't been that much progress from there. So I hope that we can get it done at some point. So yeah, if I will reach out to Nadja and follow up with her about this.
Just with respect to both issues, if you can hear me, hopefully you can hear me. You know, I think having those thresholds that we were the mindset of to give some sort of prescribed analytical plan, but I think that makes a lot more sense that would be all data are retained and make decisions on the back end. So that's with respect to motion thresholds. And Philippe, what was the second point? Forgive me. The carpet plots.
The carpet plots, yeah.
Yeah. So yeah, QC is a little different. I mean, in fact, you know, if we're looking way down the road, having some sort of semi-automated, automated QC procedures is certainly something that we're talking about, something we've written into a grant recently to help everybody. I think, I suppose, and I'd like to hear everybody else's thoughts, you could treat that similarly to your other issues of motion. Do you keep the data and then decide on the back end? If you were going to do that with motion, we have it very well documented. You can always go look. We would also need a very a formalized procedure for QC so that you can, if you're not going to be cutting scans on the front end, that on the back end you can make those decisions based on some sort of a group strategy. So motion makes more sense to me. QC is a little more, I balk a little more there on how to proceed depending on the procedures in place. So again, looking down the road, we would love to have implemented, and depending on what we're talking about QC, I assume we're talking about what you're specifically the carpet plots there, but there's a lot of different levels of QC where we can have checks. We would like to have something that's a little more automated implemented. So not sure if that helped the conversation or answered anything, but.
No, it helps a lot. I think the motion topic is kind of the tip of the iceberg that can be formalized in the first place. So I agree. The other topics also much depend on the task. If you only think of the an imaging stress task where people are activated a lot, or kids, as mentioned, that you need this flexibility. So there's no pressure or fancy ideas to get this into a formula in the next weeks.
Yeah, and also, I mean, I think we all agree, I think, that there is no best strategy for everybody for this threshold. So it's very much dependent on your data. So we can run tests, obviously, and we can easily do it, but it will very much depend on your population. So yeah, there is no good solution, but the good news is, like, as we said, it's like done at the end, so we can easily like readjust it. So for your own group, you can use a threshold that is that makes sense with your population. If you have like children, if you have like, for example, in anorexia, they don't move a lot, and so we don't have this issue, but like in ASD, they do. So we have to adjust those thresholds. But we wanted to have a general threshold of this 0.5, more like for the cross-disorder analysis. But then, for example, yeah, you can adjust. I like the idea of the percentage of frames that you mentioned, Greg. So we could think about adding that. But yeah, we could have like another meeting only about that, as it has been like suggested at some point, with like data that we could look at, comparing this threshold for several data sets. But yeah, I think there is no good solution. Okay. So we can move to the next point if there is nothing else to say about that. Okay, so another topic that people disagree on is atlas coverage. So until now, we were recommended 0.8. So we've been running, our students have been running many tests using different atlas coverage thresholds. And we saw that if you use the 0.8, it's a disaster. Your like beta map can be like almost empty. Just like some region, if you have one subject that is not covered for this region at 80%, then you lose the entire thing. So it's kind of a disaster at the end for case control analysis. So we discussed with several people in the field, and we ended up thinking that the best solution would be to do a proper visual QC to deal with this type of issues like cropping that you can easily detect when doing a visual QC. And then using this threshold of 0.5 and considering that the smoothing will help. So that was our recommendation, to not end with like empty matrices for your beta maps. So yeah, again, open to discussion with you, but that was our new recommendation for atlas coverage.
Yeah, Clara, this is great. And I think what really goes along with that, what you just said, Clara, is having a strong QC on the back end, right? So visual. So, but in this, I do think having a recommendation as opposed to the motion, I really, this is an important group discussion, because perhaps that may be less dictated. But I think in this case, if you don't have something a little more liberal in the front end, then you have no data on the back end. So I think having something like 0.5 coupled with strong QC makes sense.
Oh, sorry, I was muted. Anyone else want to say anything about atlas coverage?
Yeah, so I feel like, are you talking about seed coverage for specific seeds or total atlas coverage?
I mean, it's per seed, per region.
Right. So the issue here is that some people are going to care about some of the seeds and other people are not going to care about some of the seeds. You know, like a lot of people are going to get very poor coverage in cerebellar regions, and they're not going to care very much.
Yes.
So I think reporting on it, having quantitative reporting on it, and then suggestions, like if we have suggestions for here's what you would eliminate, that based on what you care about, that would be useful.
Yeah, also here our experience for Enigma was that we just have to go back and forth to see the data. Because if you just take everything that's available in all scans, you only have half the brain left. So it's indeed, I agree, dependent on your research question. If you care about OFC, well, then good luck. But if your question is just about visual cortex or so, yes, then you can be quite stringent. But also here, I would just log it and not make any decisions up front, because it's really dependent on what you can permit on the basis of your data set.
I think maybe also as a general comment, the problem is when we, for example, look at cerebellum, right, is that many technicians, when they do these scans, they don't really care that much about where they put the field of view. So the question is kind of, then do you want to use this region of interest in the cerebellum and look at connectivities to that specific region, even though there's really bad signal for that region only available? So I think this is also a data cleaning question. And so what HALFpipe does, it outputs a JSON file that contains all the kind of percent coverage regions for every single atlas region. And so that means, again, this filtering is possible on the group level. So that means we just run everything. And then if you want to be more stringent because you care about good cerebellar coverage, because that's part of your research question, then you can filter later. But what I would recommend is to look into statistical models that can actually handle this missing data, like, for example, to use Bayesian models or multiple imputation models that kind of use this missingness and handle it statistically, because then hopefully we can work around these imaging problems. Because in essence, it is just missing data. It is not missing at random, but it is missing data. It can be handled by existing statistical methods. No need to exclude whole parts of the brain, hopefully.
Yeah, I fully agree. It's basically like a topic close to the previous one, but what's great is like HALFpipe, as just Leah said, generate for you like one file per region, per subject, so you can adjust based on what you're interested in. But yeah, and indeed, it very much depends on the field of view they used for the acquisition. So you can be always like cropped on the top and the cerebellum. I mean, you all know that. So yeah. But yeah, we wanted to at least reduce 0.8 because we think it's very stringent. And yeah. Anything else before moving to the next topic? Also, we could have like additional more like methods meeting where we only talks about those aspects. Here it's a mix between news, updates, and like details. So we could have like meetings that are dedicated to discuss results that we are all testing on our own data sets, so that could help addressing these things. That's the goal. Is more like, because we did not do any updates since 3 months, we wanted to have a general meeting. But I guess the following ones are going to be more specifics. And we could like do a survey of the things you want to discuss. It would be a solution. Okay, so the next topic was again about like the different denoising methods. So just a reminder, we asked you like, what is a good denoising method for you that we should use? The answer was for experts, the simple one, an ACOM core. So we also asked you about GSR, and as expected, a third of usage should not be done, 2/3 said it's required or provides benefits. So we decided it was quite easy to decide that we should do both to accommodate everybody. So that's how we ended up to those guidelines that we presented to you like a while ago with Leah, with like the pipeline one between being ACOM core to be able to compare what has been done by the OCD and PTSD and MDD groups who already published paper, motion parameters and scrubbing, and the same with GSR, so with and without GSR. So that's what we recommended. We still recommend that. We did not change that, but we just wanted to talk a bit more about that. So Pierre-Lune worked on this figure from a paper published by Outing Wang that is I will share the slide that is here. I will share the slide with you. Just wanted to say that you can rank your different denoising method based on the quality of the denoising, which is in the y-axis. So it's a ranking, 1 being the best and 10 being the worst. So for example, if you run nothing, the baseline, just like high-pass filtering, you don't have a great denoising method output. And on the x-axis, you have the loss of the degree of freedom that you lose when running your denoising methods. So Pierre-Lune plotted them to be able for us to help us with the decision. I mean, we already looked at it, but we wanted to quantify it better. That's done based on the old version of fMRI prep, but doesn't matter. So what you can take from this graph, so here you have like what would be the best method, meaning you don't lose any degree of freedom and you have a great denoising. So that does not exist, obviously. That's the worst method. Pierre-Lune plotted all these methods. So for example, the simple plus GSR and simple meaning motion parameters and CompCore are quite a good intermediate choice to have a good denoising and to not lose too much degree of freedom, too many degrees of freedom. So we concluded from that if you have like short time series that you cannot afford losing a lot of degree of freedoms, it's a good choice. While if you have like long time series, you can do scrubbing, and scrubbing is better to denoise your data. So it's more like we want to rerun some tests on other data sets. That's an analysis that have been already done. So we are doing new analysis on that. But it's more to show you like when you have these different pipe outputs, like how to pick the one you want to report. And also it will help us for the multiverse analysis to compare these different outputs. But yeah, so that was my general overview of that. Maybe Lune, you want to be more specific than me, because that's you who did that.
So, I mean, I think it was very specific, just maybe to complement a little bit. So those analyses were generated in a reproducible way, the paper, I mean. So we're trying to actually kind of like rerun everything with the HALFpipe upcoming version. So we can really track if that new version behaves with a different strategy, like what we observed a long time ago, with slightly different code. So I think, at this stage, those are internal tests running in the team. Down the road, we are hoping to be able to share some kind of like reproducible report that we can use in the development cycle of HALFpipe to make sure that our common understanding of the behavior of those methods remain current and applies to whatever new versions we have.
Those data are fantastic. I hadn't seen those yet. Luna, can you just tell me, a long time series, is that 10 minutes or more? And then the short, is that 5?
There is no cutoff, like you have to.
Okay, I was just.
So the stabilization of correlation coefficient is around 10 minutes, where you see the most benefits. And so if you have more than that, and depending on how much attrition you have with QC, with FD, so it's going to depend on how much motion you have in population, I think you can apply describing safely.
Right.
And if you're not in that regime, every data point counts, and maybe you want to switch to a method that preserves this more the degrees of freedom, knowing that you're going to be left with more motion, which you can still try to model at the group level, right? So it's not like it's you're leaving those biases in the data as such. It's addressed both at the denoising level and at the group modeling level. You can do both. So if I were to give a single threshold,
I don't want to pin you down. Yeah.
10 minutes. If you can remain in that regime, I think it's fine to go for the more aggressive denoising.
That makes
But
sense.
when
With
you start
scrubbing in particular.
below that, this becomes a concern, well-documented issues on reproducibility of your matrix.
So
I also have a short question on this great plot. So the y-axis, could you explain us a bit more about how you calculated this? It says it's a rank. So
yeah,
would you also be able to plot the absolute values to get a more?
Sure. So Clara, I don't know if you want to show figure 19. And so the person who led that work is Dr. Aoting Wang, who's on the call today. She's doing her postdoctoral fellowship with me, but she's also a core NeuroiLearn developer. So those methods were integrated in NeuroiLearn a little while back. There's several metrics we looked at. We tried to replicate previous benchmarks, CIRIC in particular, CIRIC and colleagues. So that figure is a lot more complicated than the graph, but what you have is 2 different data sets and 2 different versions of fMRI prep. So that's why there's 4 bubble graphs. And if we zoom in conceptually, because I cannot zoom in real life, on one of the graph, let's say the top left, each line is a quality metric. So the very first line is the preservation of degrees of freedom, which is obviously super duper important. And the other ones are various ways you can try to get at quality of noising. Okay. So I find those plots really hard to read. So that's why I ended up averaging all the rows, except for the first one, to make like a just like XY plot, because I think that's the main takeaway. So when you're high in the ranking on the first line, you're typically lower on the other lines. So there's sort of like negative correlation between the first line and the other lines, which is what Clara was presenting. But then if you want to really zoom in certain quality metrics, I think there's a degree of convergence among them. So the 3 lines like QCFC, DMFC, essentially try to look if there's an association between your motion and your connectivity metrics at the level of single, pairs of regions. They do that a little bit differently. There's an absolute quantification, there's a significant testing, there's something where you also try to take spatial distance into account. So that's why you have 3 variants. So that would be QCFC significance, QCFC median, and DMFC. And those metrics largely converge. And then the last line is the modularity of the connectome. Like, do you really see networks really nicely? And it's a little like more of a contentious metric. It's wishful thinking. You want to see the network, so you preprocess your data, so you see the network type of deal. But it also, to a large degree, converge with the other ones. So in the XY plot that Clara showed and that I wish we had put in the paper because it's much easier to read, we're averaging all those quality metrics. And why do we use ranking? It's because those metrics live in completely different scales. So it makes it hard to just average them.
That would also be my question, because if you rank it, it automatically gets the best and the worst. Yeah. While one scale, it may just not matter that much. There's little variance, whereas in some scales, it might be have a huge effect. And this would be difficult to see here.
That's correct. I don't think it's perfect. We had worked with the absolute. We found it harder to deal with. Maybe we should have like kind of normalized by mean and variance, for example, per metric. So that would have, put everybody on the same scale, but also address your concern, Guido, that if there's small change, you may not want to,
now, let's say if the degrees of freedom is just 1, 2, and 3, and 1 to 9, then you have them all in the rank, but it essentially doesn't matter that much. So the same goes for all the others.
I'm taking it as a note. It is a good idea. I think we need to get a better plot, is what I take away. I'm not sure we need to solve this on the large group call. But yeah, those are great suggestions. Thank you.
I think I will wrap up real quick because it's been 1 hour. So I just want to give to just say a few words about the rest, and then we can go back to that if some have time to stay. But yeah, that was a way to summarizing these different motion denoising methods that's here. So the one we picked are those one, but we wanted also to show you that to explain the reasoning behind. And for the grant we wrote, we would like to do multiverse analysis to compare more deeply these different denoising method. We don't want everybody to do it. That's not the research question of each working group here, but we would like to do that. That's why we still recommend the three first pipeline that are like just the screenshot from the current HALFpipe implementation. But for the groups who are interested to dive into those things better, we would recommend to add 2 additional pipeline, which is just like the simple one with only motion parameters and no scrubbing, and the other one is basically the same but with GSR. So we will, I don't have time to dive into that more. Oh wow, what's going on? Okay. I don't know, Sarah, if you can remove that, but okay. So we will, we have been working on the guidelines. They are almost ready. We were waiting because we wanted to have the fMRI prep new version before releasing the version of the new HALFpipe plus the guidelines together. But yeah, we will have that. It's quite similar to what has been already, I mean, it will look similar to what has been done by the OCD and PTSD MDD groups. So hopefully we will have this document next week or something like that. We are preparing a preprint with Leah, Perlune, Ilya, and Frank. The last things, like we submitted this ROI and R1 in February, so we don't have time to dive into that, but just like the aim one of it was to optimize the fMRI workflow for speed, accessibility, and sensitivity analysis. So what it means is like the first, we want to have to implement deep learning methods like deep prep to accelerate the processing of the analysis, the pre-processing of the images. The second aim to develop a semi-automated QC because that's one of the main issue in that we have here, especially in ENIGMA, because like some groups are happy to run HALFpipe, but they don't have someone that has time to understand how to properly do the visual QCs. So, and we cannot share the data for QC. So that's a major problem for ENIGMA. So that's why we put it here. And the last one is to compare the different denoising method doing multiverse analysis. So that's what we were talking about with Perlune. So yeah, and then we have no time for questions. We have our next meeting in exactly a month. If you want to add someone to the mailing list, please email Sophia. If you want to leave the mailing list, please email Sophia. So yeah, just email Sophia. And yeah, and we have some update from Frank, but I think for next call, it will be better. So yeah, if anyone has questions,
definitely. Yeah.
If people have to leave, that was I just wanted to update the general things, but yeah, we can go back to that discussion. I also agree that's the most interesting thing. Well, thanks so much for everyone for joining, and we will share the slides and hopefully the documentation next week. And we will email you when the new LTS is ready to be used. It's ready to be used right now, but like it's better if you still wait for us to test stuff. Thank you very much.